Okay, let's dive in. It's pretty fundamental, isn't it? This human need to, well, organize everything we see, especially the living world.
Absolutely. And it goes way back. Long before, fancy labs and genetics, people were sorting things out instinctively.
Just based on what they could see, right? Like, is it a plant or an animal? Does it move?
Exactly. Think about Aristotle. His categories, plants into trees, shrubs, herbs, animals, based on whether they had red blood, seem basic now.
Yeah, pretty simple groupings.
But it was a start, a move towards being systematic. And honestly, it was practical too. Knowing what you could eat, what might hurt you, survival stuff.
Makes sense. But then, well, technology happened. Microscopes changed everything. Suddenly, those simple bins, they weren't quite cutting it anymore.
No, not at all. Linnaeus's two kingdoms, Plantae and Animalia, was huge for its time, don't get me wrong, super influential.
But it started showing cracks pretty quickly, yeah. When we
Right.
found things that just didn't fit neatly.
Totally. Where do you put bacteria? Or fungi? They're not really plants, not really animals in the way Linnaeus thought. And the whole prokaryote versus eukaryote thing.
Right, that fundamental difference in cell structure. No nucleus versus having one.
And how they get food, photosynthesis, absorbing stuff. The two kingdom model just couldn't stretch that far. It lumped too many different things together.
So we needed a better system, something that reflected, well, the actual biological diversity we were discovering.
Exactly. Which brings us to R.H. Whitaker in 1969. His five kingdom classification was, well, a game changer.
Monera, Protista, fungi, Plantae, and Animalia. That's the one most of us probably learned.
And the key thing was why he divided them that way. It wasn't just looks, he looked deeper.
Like cell structure.
Cell structure, definitely. How complex the organism's body was, single-celled, multicellular, how it got nutrition.
Autotroph, heterotroph.
Right, how it reproduced. And crucially, he brought in phylogeny, evolutionary relationships.
Ah, the family tree aspect. Trying to group things based on shared ancestry. That feels much more, I don't know, fundamental.
It was a huge leap towards understanding how life's actually connected. Now, science doesn't stand still, of course.
Right, people talk about the three-domain system now, splitting Monera.
Exactly. Leading to maybe 6 kingdoms or more, depending on who you ask. But understanding Whitaker's 5 gives you a really solid foundation for grasping, the major branches of life.
Okay, so for this deep dive, we're focusing mainly on those 5. Monera, Protista, fungi, Plantae, Animalia. Although we'll touch on Plantae and Animalia less since they're more familiar.
Sounds like a plan. It's a great framework. Where should we start?
Let's kick off with Kingdom Monera, the prokaryotes. No nucleus, remember.
Right. And this kingdom is basically bacteria. They are, well, they're everywhere, absolutely everywhere.
Soil, water, air, inside us, on us, hot springs, deep sea vents.
You name it. They are the masters of adaptation and incredibly abundant, just staggering numbers.
And they come in different shapes too, don't they? I remember seeing diagrams.
Oh, yeah. You've got the spherical ones, coccus, rod-shaped bacillus, comma-shaped vibrio, and spiral ones, spirillum. Basic forms, but lots of variation within them.
Simple shapes, but metabolically. Wow, they do everything.
That's the amazing part. Their metabolic diversity is huge. Some are autotrophs, making their own food.
Like photosynthetic ones,
Yeah.
using sunlight.
Yep, some do that, but others are chemosynthetic. They get energy from inorganic chemical reactions, oxidizing things like ammonia or sulfur. No sunlight needed.
That's wild. Life running on chemical energy alone.
Yeah.
Gives you hints about early life, maybe.
Potentially, yeah. And then the vast majority are heterotrophs. They consume organic matter, just like us, but in simpler ways.
So decomposers, parasites, symbionts, they cover all the nutritional bases?
Pretty much. And within Monera, you have these two major groups, the archaebacteria and the eubacteria.
Archaebacteria, those are the extremophiles, right?
Yeah.
The tough ones.
Exactly. They live in places most things couldn't survive. Extreme salt halophiles.
Oh.
Really hot, acidic water, like in hot springs, thermoacidophiles. And then there are methanogens, living in marshy, oxygen-free places, or even in the guts of cows and other ruminants.
Ah, so that's where the methane comes from. It's the archaea doing their thing?
That's them. And they can handle these conditions because their cell walls are structured differently from other bacteria, unique chemistry.
Fascinating adaptations. So what about the eubacteria, the true bacteria?
All right, eubacteria. They have rigid cell walls, usually made of peptidoglycan. That's a key difference. And if they move, they typically use a flagellum, a whip-like tail.
And this group includes some really important players. like cyanobacteria.
Oh, definitely. Cyanobacteria, sometimes called blue-green algae, though they're not true algae, they're photosynthetic champions.
They have chlorophyll A, just like plants.
They do. So they perform oxygenic photosynthesis. They can be single cells, form colonies, or make long filaments. Found in water, moist soil, pretty common.
Don't they sometimes form those blooms in polluted water?
They can, yes,
Mhm.
often covered in a gelatinous sheath. And some are incredibly important because they can fix atmospheric nitrogen.
Taking nitrogen gas from the air and turning it into something usable.
Exactly. In specialized cells called heterocysts, think of organisms like nostoc or Anabaena. They're natural fertilizers, basically.
Wow. Okay, so we have photosynthetic ones, nitrogen-fixing ones. What else in eubacteria?
Well, you also have chemosynthetic eubacteria, similar to some archaea. They play vital roles in recycling nutrients, nitrogen, phosphorus, iron, sulfur, by oxidizing inorganic stuff, essential for ecosystems.
The unsung heroes of nutrient cycling.
Definitely. But the most abundant group overall are the heterotrophic eubacteria.
The decomposers and, well, the ones that interact with us more directly.
Right. Huge role as decomposers, breaking down dead things, but also vital in things like making curds from milk, producing antibiotics.
Penicillin came from a fungus, but other antibiotics.
Many come from bacteria, yeah, like streptomycin, and they help fix nitrogen in the roots of legumes like peas and beans.
But there's a downside too, right? Pathogens.
Unfortunately, yes. Many diseases are caused by heterotrophic bacteria. Cholera, typhoid, tetanus, even plant diseases like citrus canker. It's a mixed bag.
How do they reproduce mostly? Just splitting.
Primarily by binary fission, yes. One cell divides into two identical ones. Pretty straightforward.
But they can handle tough times.
They can form spores, yeah, really resistant structures to wait out bad conditions. And they even have ways of exchanging genetic material, a sort of primitive DNA transfer. Not true sexual reproduction, but it allows for variation.
Okay. And one last weird one in Monera, mycoplasma.
Ah, yes. Mycoplasma are kind of the odd ones out. Their main thing, no cell wall at all.
That's weird for a bacterium.
It is. They're also incredibly tiny, the smallest known living cells. And they can survive without oxygen. Many are pathogenic in plants and animals, too.
So Monera, prokaryotic, incredibly diverse, metabolically versatile, and absolutely foundational for life on Earth.
You got it. A whole world in tiny packages.
All right, where to next? Kingdom Protista, the eukaryotic starting point.
Sounds good. Protista. Yeah, this kingdom is often described as, well, a bit of a mixed bag. Or a grab bag.
Because it's basically all the eukaryotes that aren't plants, animals, or fungi.
Pretty much. It's defined more by what it isn't. But the key is, they are eukaryotes. They have a propyl nucleus, membrane-bound organelles. The works.
And mostly single cells.
Mostly single-celled eukaryotes, yeah. Though some form colonies or simple multicellular structures. It includes groups like chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans.
Quite a variety. Where do you find them mostly?
Primarily aquatic, freshwater, marine environments. They're key players in aquatic ecosystems. And you can think of them as a crucial link.
A link? How so?
They bridge the gap between the simpler prokaryotes and the other complex eukaryotic kingdoms. You see features in protists that hint at the ancestors of plants, animals, and fungi.
Interesting, like having flagella or cilia for movement.
Exactly, and their cell structure is clearly eukaryotic. They can reproduce asexually, but many also have sexual reproduction involving cell fusion and zygote formation, early steps towards complex life cycles.
Okay, so let's break down some of those groups. Chrysophytes, what are they?
Chrysophytes include diatoms and golden algae, or desmids, mostly found drifting in water, plankton.
Plankton, so tiny floating organisms.
Microscopic, yeah. And crucially, most are photosynthetic. They are major producers, especially in the oceans, the base of many food webs.
Diatoms, they're the ones with the cool shells, right?
That's them. Their cell walls are embedded with silica, forming these intricate overlapping halves, like a little soapbox or petri dish. Beautiful structures.
Made of silica like glass.
Essentially, yes.
Yeah.
And when they die, these silica shells sink and accumulate over millions of years.
Forming?
Diatomaceous earth. That gritty stuff used in filters, polishing agents, even toothpaste sometimes.
Wow. From tiny plankton shells. Okay, next group. Dinoflagellates.
Dinoflagellates. Mostly marine, also photosynthetic. They come in various colors, yellow, green, brown, blue, red, depending on their pigments.
And they have a distinct structure, too.
They do. They have stiff cellulose plates forming their cell wall, like armor, and characteristically, two flagella.
Two. How do they work?
One lies in a groove longitudinally, and the other sits in a transverse groove around the middle. Their beating makes the dinoflagellate spin as it moves.
Spinning swimmers. But aren't they also responsible for red tides?
Ah, yes. Some species, like Goniolax, can multiply incredibly rapidly under certain conditions. These blooms can turn the water red.
Now that's bad news, right?
Often, yes.
Yeah.
They can produce potent toxins that kill fish and other marine life. And if shellfish consume these toxic dinoflagellates, they become poisonous to humans.
Scary stuff. Okay, moving on. Euglenoids, like Euglena.
That's the classic example, Euglena. Euglenoids are mostly freshwater organisms, often found in stagnant water.
And they're a bit different structurally.
Yeah, instead of a rigid cell wall, they have a flexible protein layer, called a pellicle, just inside the cell membrane. Allows them to change shape a bit.
Flexible. And
Yeah.
nutritionally, they're adaptable.
Very. They are mixotrophic. If there's sunlight, they photosynthesize. They have pigments very similar to higher plants, actually chlorophyll A and B.
Another connection.
Right. But if they're kept in the dark, they can switch modes and act like heterotrophs, absorbing nutrients or engulfing smaller organisms.
Best of both worlds, then. Okay, now for the really weird ones. Slime molds.
Haha. Yeah, they are definitely unique. Slime molds are saprophytic protists. They feed on decaying twigs, leaves, organic gunk.
Saprophytic, so they absorb nutrients from dead stuff. But their life cycle is strange.
It really is. Under good conditions, they exist as this multinucleated mass of cytoplasm called a plasmodium. It can creep along like a giant amoeba, engulfing food particles, can get quite large, actually.
Giant creeping blob.
Okay.
But when conditions get tough dry or food runs out, the plasmodium changes. It differentiates and forms fruiting bodies. Fruiting bodies, like fungi.
Sort of analogous, yeah. These structures bear spores at their tips. And the spores are really resistant.
So they can survive hardship and then spread.
Exactly. The spores get dispersed, usually by air currents, and if they land somewhere suitable, they germinate and start the cycle over.
Wild. Okay, last group in Protista, the protozoans.
Protozoans, these are all heterotrophs. They live as predators or parasites, and they're generally thought of as being the primitive relatives of animals.
Makes sense. Heterotrophic, often mobile, and they're grouped by how they move.
Primarily, yes. First, the amoeboid protozoans. Like amoeba itself, they move and capture food using pseudopodia.
False feet, those extensions of the cell.
That's right, just projecting their cytoplasm. Found in freshwater, seawater, moist soil, some marine forms have silica shells, and some are parasitic, like entamoeba, which causes amoebic dysentery.
Got it. Second group.
Flagellated protozoans. As the name suggests, they use flagella for movement. They can be free-living or parasitic.
Parasitic examples.
A notorious one is trypanosoma, which causes African sleeping sickness, transmitted by the tsetse fly.
Nasty. Okay, third group.
Ciliated protozoans. These guys are covered in thousands of tiny hair-like cilia.
Cilia, like tiny oars.
Exactly. They beat in coordinated waves, allowing for pretty rapid movement. They're aquatic. And the cilia also help sweep food particles towards a specific opening called the gullet.
Like paramecium, the slipper-shaped one.
Paramecium is the classic example, yes. Very active movers.
And the final protozoan group.
Sporozoans. This group is diverse, but they all have an infectious, spore-like stage in their life cycle, and they are all parasites.
Spore-like stage, like the slime mold.
Different kind of spore, really. More like an infectious stage for transmission. The most infamous sporozoan is Plasmodium.
Malaria.
The malarial parasite, yes. Complex life cycle involving mosquitoes and humans. Huge impact on human health globally.
So Protista, really diverse.
Huh.
Single-celled eukaryotes doing all sorts of things. Photosynthesis, predation, parasitism. Quite the kingdom.
It truly showcases the explosion of possibilities that came with the eukaryotic cell structure. A lot of evolutionary experimentation happened here.
Right. Okay, let's move on to kingdom fungi. Another group that isn't plant or animal.
Correct. Fungi are a unique kingdom of strictly heterotrophic organisms, and wow, the diversity in form and habitat is incredible.
Yeah, you see them everywhere. Mold on bread, mushrooms popping up after rain, toadstools.
Rotten fruit, yeast for baking and brewing, but also less obvious ones like microscopic parasites causing plant diseases
It's like wheat rust. I've heard of that.
caused by Puccinia, a fungus. And conversely, some fungi give us valuable things like antibiotics, Penicillium is a prime example.
Right, from the Penicillium mold, so they're cosmopolitan, found everywhere.
Pretty much worldwide. They especially love warm, humid places. Makes sense for decomposition.
And structurally, they're mostly made of filaments, except yeast.
Exactly. Yeast are typically unicellular, but most fungi are filamentous. The individual filaments are called hyphae, long, slender, thread-like structures.
And a mass of these hyphae is.
The mycelium, that's the main body of the fungus, often hidden in the soil or wood or whatever it's growing on.
And these hyphae can be different types.
Yes. Some fungi have conocytic hyphae. These are like continuous tubes filled with multinucleated cytoplasm, no cross walls or septa, allows for rapid flow.
Efficient nutrient transport, maybe?
Seems likely. Other fungi have septate hyphae, where there are cross walls dividing the filament into individual cell-like compartments, though the septa often have pores.
And their cell walls aren't cellulose like plants.
Nope. Fungal cell walls are primarily made of chitin, a tough polysaccharide, same stuff found in insect exoskeletons, and other polysaccharides too.
Chitin, okay. And they're all heterotrophic. How do they feed?
They secrete digestive enzymes out into their environment. These enzymes break down complex organic molecules, and then the fungus absorbs the simpler nutrients through the hyphae walls.
So external digestion.
External digestion, yeah. And based on what they digest, we categorize them. Saprophytes absorb nutrients from dead organic matter, the great decomposers. Parasites depend on living plants or animals for their nutrients, often causing harm.
And symbionts.
They live in close association with other organisms, often mutually beneficial. Two key examples: lichens,
Fungus plus alga.
or cyanobacterium, yes, and mycorrhizae.
Fungus plus plant roots.
Right. Hugely important for nutrient uptake for many, many plants. The fungus helps the plant get phosphorus and other minerals.
Okay, how about reproduction in fungi?
They can do it all. Vegetative reproduction is common, just fragmentation of the mycelium where a piece breaks off and grows into a new fungus, or fission and budding in yeasts.
Simple cloning. Asexual.
Lots of asexual reproduction via spores, different kinds. Conidia, sporangiospores, zoospores, which are motile flagellated spores. These are usually for rapid dispersal.
And sexual reproduction.
Yes, that happens too. Also involving spores, oospores, ascospores, basidiospores. These sexual spores are typically produced in distinct structures called fruiting bodies like a mushroom.
The mushroom is the reproductive structure then.
It's the fruiting body designed to produce and release sexual spores, yeah. The actual fungus is the mycelium underground.
And the sexual cycle has steps?
3 main steps. First, plasmogamy, the fusion of the cytoplasm from 2 parent mycelia.
Okay, cytoplasms merge.
Then comes karyogamy, the fusion of the 2 haploid nuclei. This forms a diploid zygote nucleus.
Nuclei merge. Now it's diploid.
Right. And finally, meiosis occurs in that zygote nucleus, reducing the chromosome number back to haploid and producing the sexual spores.
Back to haploid spores, but didn't you mention a weird stage before?
Ah, yes. In some groups, particularly Ascomyces and Basidiomycetes, karyogamy doesn't happen immediately after plasmogamy.
There was a delay.
Yeah. You get this intermediate stage called a dekaryon. The cell contains 2 separate haploid nuclei, one from each parent. We represent it as N plus N.
2 nuclei just hanging out together in the same cell. For how long?
Can last quite a while in the life cycle. Eventually, those nuclei will fuse karyogamy just before meiosis.
Interesting complexity. So how are fungi classified based on these reproductive things?
Largely, yes. The morphology of the mycelium, septate or coenocytic, the mode of spore formation, asexual and sexual, and the type of fruiting bodies formed are the main criteria used to divide the kingdom into different classes.
What are the main classes?
Traditionally, Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Though Phycomycetes isn't really used as much formally now, it's a useful grouping to start with.
Okay, let's look at Phycomyces first. The lower fungi.
Yeah, often found in aquatic habitats on decaying wood in moist places, or sometimes as obligate parasites on plants.
Obligate parasites, meaning they have to be parasitic.
Right, can't survive otherwise. Their mycelium is typically aseptate and coenocytic, those continuous tubes.
How do they reproduce?
Asexually by spores, formed inside a structure called a sporangium. These can be motile zoospores, if aquatic, or non-motile aplanospores. Sexually, they form a zygospore after fusion of gametes.
Zygospore. Thick-walled resting spore.
Usually, yeah. The gametes fusing might look identical, isogamous or different, anisogamous or oogamous.
Yeah.
Examples include common bread mold, Rhizopus or Mucor, also albugo, which causes white rust on mustards.
Got it. Next, Ascomycetes, the sac fungi.
Sac fungi, exactly. A huge and diverse group, mostly multicellular, think Penicillium, Aspergillus, Neurospora, but also includes unicellular yeasts like Saccharomyces.
So baking yeast is an ascomycete. What do they do ecologically?
All sorts. Saprophytic decomposers, parasites, some are coprophilous, grow on dung. Some form mycorrhiza or lichens.
And the structure.
Mycelium is branched and septate has those cross walls. Asexual reproduction is typically by spores called conidia, which are produced externally on specialized stalks called conidiophores, not inside a sporangium.
Conidia. Okay. And sexually, why sac fungi?
Because their sexual spores, called ascospores, are produced inside a sac-like structure called an ascus. Usually there are 8 ascospores per ascus.
8 spores in a sac. And these asci are...
Often grouped together within a larger fruiting body called an ascocarp. Various shapes. This group includes Neurospora, famous in genetics research.
Why famous?
Easy to grow, short life cycle. Ordered spores in the ascus made mapping genes easier. Also includes edible delicacies like morels and truffles. And less desirable ones like claviceps, which causes ergot on rye.
Ergot. That's poisonous, right?
Yes, and hallucinogenic, source of LSD precursors. Okay, next up, Basidiomyces.
The club fungi. This is mushrooms, right?
Mushrooms are the most familiar example, yes, but also bracket fungi you see on trees, puffballs, and the parasitic rusts and smuts that affect crops.
Where do they grow?
Soil, logs, tree stumps as decomposers or as parasites on plants. Their mycelium is also branched and septate.
How do they reproduce? Asexual spores common.
Asexual spores are generally not found. Vegetative reproduction by fragmentation is common, though. Sex is the main event here.
And it involves clubs?
Sort of. The defining feature is the basidium, a club-shaped structure where karyogamy, nuclear fusion, and meiosis occur.
Okay, the dikaryotic stage leads to the basidium.
Right. Typically, meiosis in the basidium produces 4 haploid nuclei, which then develop into 4 basidiospores. Crucially, these basidiospores are born externally on little stalks on the basidium, exogenous spores.
Outside the club, not inside like the ascus.
Exactly. And these basidia are usually arranged on or in a complex fruiting body. The basidiocarp, the mushroom itself, is a basidiocarp.
So agaricus, the common mushroom, Ustilago, smut fungus, Buccinia, rust fungus, all basidiomycetes.
Correct. No obvious sex organs usually. Plasmogamy often happens just by fusion of regular vegetative hyphae from compatible mating types.
Complex stuff. Okay, one more group. Deuteromycetes, imperfect fungi.
Imperfect fungi, yes. This is basically a holding category. It includes all the fungi where we've only observed asexual or vegetative reproduction. We haven't found or identified a sexual stage yet.
So imperfect means our knowledge is incomplete.
Precisely. They reproduce asexually, typically by conidia, just like many ascomycetes. Their mycelium is septate and branched.
What kind of fungi end up here?
Many are saprophytes or parasites. Important decomposers involved in mineral cycling. Examples include Alternaria, Calatotricum, Trichoderma.
And what happens if scientists do find the sexual stage for one of these?
Then it gets moved. Based on the characteristics of its sexual reproduction, it's reclassified as either an ascomycete or a basidiomycete. So deuteromycetes is kind of a temporary classification based on missing information.
Makes sense. Fungal classification is clearly dynamic.
Very much so, especially with molecular data shaking things up now, too.
Okay, so we've covered Monera, Protista, fungi. What about those other things? Viruses, viroids, prions, and lichens. Whitaker didn't include them.
No, they're outside the 5-kingdom system for specific reasons. Viruses, viroids, and prions aren't cellular. They lack the basic machinery of life that cells have.
A cellular, not made of cells.
Right. And lichens. Well, they are cellular, but they're not a single organism. They're a symbiosis.
A partnership. Okay, let's tackle viruses first. We hear about them all the time, especially lately.
Indeed. Viruses are fascinating because they blur the line between living and non-living. Outside a host cell, they're inert, like tiny crystals of chemicals.
Inert, but infectious.
Exactly. Once they get inside a suitable host cell, they hijack the cell's machinery, its ribosomes, enzymes, energy to make more copies of themselves. They basically turn the cell into a virus factory.
Often killing the host cell in the process.
Often, yes. That's why they're called obligate intracellular parasites. They absolutely must be inside a cell to replicate.
Who figured these things out?
Early work by Ivanowski on tobacco mosaic disease showed something smaller than bacteria was causing it. Beijerinck called it a contagious living fluid. Then Stanley managed to crystallize tobacco mosaic virus, showing its particle nature.
So what are they made of? Just genetic material and protein?
That's the core structure. A nucleic acid core, either DNA or RNA. never both surrounded by a protein coat called the capsid.
DNA or RNA. That's a key difference in cells, which always have DNA as the main genetic material and use RNA for various roles.
Correct. And the type varies. Plant viruses usually have single-stranded RNA. Animal viruses can have single or double-stranded RNA or double-stranded DNA.
And viruses that infect bacteria. Bacteriophages.
Bacteriophages, or just phages, usually have double-stranded DNA. They often look like little lunar landers.
And the protein coat, the capsid.
Made of smaller protein subunits called capsomeres. Its job is to protect the nucleic acid and help the virus attach to and enter a host cell.
And they cause a ton of diseases, right?
Yeah, in humans.
Yeah.
Mumps, smallpox, herpes, influenza, the common cold, AIDS. The list is long. And in plants, mosaic diseases, leaf curling, yellowing, stunted growth, major agricultural impact.
Okay. Smaller than viruses, viroids.
Even simpler. Discovered by T.O. Diener, studying potato spindle tuber disease, viroids are basically just a small circular molecule of RNA. That's it.
No protein coat, just naked RNA.
It's just free RNA, yeah.
Oh,
Much
yeah.
smaller than viral genomes. And somehow this tiny piece of RNA can get into plant cells and cause disease, likely by interfering with the plant's own gene regulation, low molecular weight RNA.
Wow. Simpler still. Now, prions. These sound really weird.
Prions are incredibly strange. They're infectious agents thought to be composed solely of protein, abnormally folded protein.
Infectious protein. No DNA or RNA at all.
Seems that way. Similar in size to viruses, but no genetic material detected. The leading hypothesis is that the abnormal prion protein acts like a template.
The template.
It bumps into normal regions of the same protein in the host, especially in the brain, and it causes them to misfold into the abnormal infectious shape. It's like a chain reaction of misfolding.
And this causes diseases.
Devastating neurological diseases. Bovine spongiform encephalopathy, BSE, mad cow disease in cattle, and variant Creutzfeldt-Jakob disease, VCJD, in humans, basically riddles the brain with holes.
Truly bizarre and frightening. Okay, lastly, the odd one out for a different reason. Lichens.
Lichens. Not a cellular, but also not a single organism. They are a classic example of symbiosis, a very intimate partnership.
Between.
A fungus, called the mycobiont, and an alga or a cyanobacterium, called the phycobiont.
Fungus and a photosynthetic partner. What's the deal?
It's mutualism. The alga or cyanobacterium photosynthesizes, providing carbohydrates food for both partners.
Okay, the photobiont makes the food. What does the fungus do?
The fungus provides the structure, the house, essentially. Its hyphae wrap around the algal cells, protecting them from harsh conditions, especially drying out. The fungus also absorbs water and mineral nutrients from the environment, which both partners use.
So they're better together.
Much better. The association is so close that lichens look and behave like single organisms. They have unique shapes and colors, totally different from either partner growing alone.
And they're useful indicators.
Very good indicators of air quality. Many lichens are extremely sensitive to pollution, especially sulfur dioxide, so if you see lots of diverse lichens growing somewhere, it's generally a sign of clean air. If they disappear, could be a problem.
Fascinating. A whole living partnership acting as an environmental monitor. Okay, so wrapping this up. We've journeyed from early attempts at classification to Whitaker's influential gangs, Monera, Protista, Fungi, Plantae, Animalia, really digging into the first three. We saw the prokaryotic world, the rise of eukaryotes, the unique nature of fungi.
And we highlighted the criteria that became important: cell structure, nutrition, organization, reproduction, and those crucial evolutionary relationships. It's about understanding the diversity and the connections.
We also touched on why things like viruses, viroids, and prions don't fit the kingdom model because they aren't cellular, and why lichens stand apart as symbiotic composite organisms.
It really shows how our understanding has evolved, moving beyond just visible characteristics to incorporate microscopic and biochemical details, and now increasingly molecular data.
Which brings us to a final thought. Science keeps moving, right? We have genomics, molecular biology tools getting more powerful all the time.
Constantly refining our view of that tree of life.
So here's something to think about. Given these advances, how might our ways of classifying life change again in the future? Are we likely to see major revisions, maybe entirely new ways of grouping organisms based on deep genetic histories that we're only just uncovering?
What currently hidden relationships or fundamental divisions might emerge as we sequence more genomes and compare them? Could the whole kingdom or domain structure be redrawn in ways we can't quite predict yet?
It's a reminder that the way we organize life reflects our current understanding, and that understanding is always growing. What surprises might the tree of life still hold?
A question that keeps biologists busy, that's for sure. Always more to discover.